bases in groups of 2, you'll get only 16 possibilities, and that's not enough either. And so let me sort of just show you like AA or AU or AC or AG, and then if you continue to GG, GU, GC, and GA, if you keep doing that, there would only be 16. So the genetic code is read in groups of 3, 1, 2, 3, and 1, 2, 3, and 1, 2, 3, and 1, 2, 3. So it's every 3 is the codon that corresponds to an amino acid. So if you read the genetic code in groups of 3, that gives you 64, so that's 64 possibilities, so that's even more than you need. And so we'll get into that a little bit later in the conversation. So the genetic code is read in groups of 3, but here's the question. What is the genetic code, and how is it translated into, in other words, what's the genetic code for glycine? Is it CAG? Is it UAG? In other words, what's the genetic code for phenylalanine? What's the genetic code for lysine? So how can you design an experiment to figure out the genetic code? In other words, crack the genetic code. It's kind of interesting, huh? So why is this relevant? Well, each protein has a unique sequence of amino acids, and that's known as the primary structure. So the sequence of amino acids, and so the genetic code in messenger RNA, and then obviously in DNA, holds the recipe for the sequence of amino acids. And why is that important? Because the primary structure of proteins ultimately determines its three-dimensional folding, and its three-dimensional shape determines its functionality. So this is hugely important. Okay, so how are we going to do it? Well, I'll talk to you again about this. And so we now know how the genetic code is read. For example, AUG happens to be a start codon. It's the start of most proteins, and it also is the codon for the amino acid methionine. And ACU is for threonine, and GGC is for glycine. Okay, so you're like, well, I'm revealing the code. Yes, I'm revealing the code, but it's showing you that we do know it. But I want to know how we determined it. That's what I'm going to talk about. And so I hope this is intriguing you. So Marshall Nuremberg, upon discovering the genetic code, gave a seminar describing the assay in which he went about doing this. And so in 1961, as it turns out, kind of curious, but he gave this presentation in Moscow, and it wasn't well attended, considering the magnitude of the discovery. Okay, one of the great discoveries of all time is telling you in biology. But perhaps it was the name. Remember this if you're ever giving a presentation or writing a book. Be careful about calling it something standoffish or intimidating. So he called it the title of his presentation, "The Dependence of Cell-Free Protein Synthesis Upon Natural and Cell-Free RNA." Apparently that wasn't just bringing in the huge crowd. But as it turns out, as we'll see, when you understand the experiment, this is actually a pretty accurate title. And so this was the seminar that cracked the genetic code. So let's get into that. And so what he went about doing was, this is a description of the experiment or the assay. So Marshall Nuremberg took some E. coli, some bacteria. Okay, so hopefully you're familiar with that. These are prokaryotes. They have one chromosome of DNA, and of course transcription produces RNA, and they have ribosomes. And that's pretty much it for prokaryotes. You have these ribosomes, and of course ribosomes are where messenger RNA is translated into protein. So this is what Marshall does. He takes the bacteria and he puts them in a blender, and he breaks them down. In other words, breaks down the cell wall and cell membrane, makes something like, so this is the extract, we're going to call it, sort of like a bacterial soup. And so you're like, well, hold on, what is that? Well, again, these are bacteria, so this is a prokaryotic cell. If you put it in a blender, you're going to break the cell walls down. You can use other techniques. You can use enzymes called lysozymes and freeze to break open bacteria. It's commonly done. But you can break the bacteria open, and then you can also add an enzyme that will degrade the DNA. And as it turns out, he went ahead and did that. So basically what the extract is cytoplasm, and you've got ribosomes. So you've got ribosomes in here. And so ribosomes, again, just to clarify this, to make sure you're following me, a ribosome, which is this guy right here, reads messenger RNA, reads RNA, and it's the one that contains the genetic code. So in other words, the ribosome is reading the genetic code in groups of 3 and attaching the amino acids together, forming peptide bonds to produce a polypeptide. So proteins are being made on ribosomes. And so, okay. So what does he do? He takes the bacteria, he breaks it down to get ribosomes in here. Okay. So these are the ribosomes. And then he adds to this E. coli extract, he adds some radioactive amino acids. So he's able to do that. Kind of clever. Okay, so let me just get into that. So these amino acids are radioactive, so they can be traced. Okay, right there. So radioactive amino acids, he's adding these. And so whatever this juice was, and we know what it is, it's a bunch of ribosomes. Whatever the juice is, the extract, it can make proteins. So in other words, you can take these radioactive amino acids and link them together and make a protein. And you're like, huh, okay, that's interesting. So this is what he does. He blenders the E. coli. He makes an E. coli extract. He adds amino acids to it, and they make protein. Okay. So what's really cool about this, though, is if you take this extract and you put it in an incubator, 37 degrees, and you let it sit in there for about a half an hour, about 30 minutes. Okay. And then you do the same thing. You add these radioactive amino acids. So there you are adding those radioactive amino acids, and then lo and behold, there's no protein. So a negative result, that's kind of weird. And so you might be thinking, I don't know, maybe it's the temperature, but if it turns out that it has nothing to do with the temperature. And what's interesting about this is that if you put it in the incubator just for 5 minutes, say, it is capable of producing a protein. But if you wait approximately 30 minutes and add these amino acids, there's no protein. So that's kind of strange. Something to think about. Okay? So it's a negative result when you let it sit around for 30 minutes. 45 minutes, no protein. An hour, no protein. 2 hours, no protein. But about 5 minutes, it's still capable of making a protein. Okay? So as it turns out, what's even more cool about this, a little more interesting, is that you take the same E. coli extract. Okay, it's the same thing. You blender the E. coli, so the ribosomes are in here, and then you're adding your radioactive amino acids to it. Okay? You let it sit for 30 minutes at 37 degrees, and you're like, well, it's not going to make a protein. Ready for this? If you then add RNA, okay, if you add RNA from a different E. coli, let me do this properly, a different E. coli. So in other words, exogenous RNA, so free RNA. If you're adding free RNA to this, guess what? It'll make a protein. So what do you make of that? It's like you thought that the extract wasn't capable of doing it after 30 minutes, but then you add RNA and it is capable of making a protein after all. What are your thoughts on that? Okay, you can pause the video and ponder this for a little bit if you felt like it. So this is a positive result. This is a little weird. But the implication of this is huge. Okay, the implication is, what's happening during the wait? What's happening between 5 minutes it can make a protein, 30 minutes it can't make a protein, but then when you add RNA, free piece of RNA from a different group of E. coli, you can make a protein. The only logical conclusion is that somehow the RNA from the E. coli that you're working with in the extract degrades. In other words, it's no longer present. And we now know that about RNA, is that it's short-lived. There's RNA aces that break down RNA very rapidly in the cell. And that's to the advantage of cells. You don't want messages around in the cytoplasm that are old. So you want to, today's news is important. Make that protein, but then get rid of that message. And so it's a way in which the cell can be efficient in terms of protein synthesis. So RNA doesn't last very long in the cytoplasm. So in the E. coli extract, RNA is broken down. So therefore, you can add all of the amino acids you want to your merry content, and there would be no protein. You need RNA. So if you add RNA to this, the ribosomes are fine. The ribosomes are fine. And so you're like, okay, what's the big deal? How's this even cracking the genetic code? This is huge. Because you know what we've just been able to do? If you can take E. coli, so what we're saying here is if you can take E. coli, make an extract, okay, let it sit for about 30 minutes, then it's basically ribosomes capable of making a protein if you give it RNA. So you have something like paramount to the fact that the E. coli extract can be, if you will, programmed to produce a protein if you give it a piece of RNA. So in other words, it's like a DVD player. It'll play whatever DVD you put in, or CD player, it'll play whatever you put in. Any RNA that you put in, it'll produce a protein. And so this is huge because, so here's the thought, that if you give the E. coli extract a simple enough RNA, now the truth be known is back here, when you added this exogenous RNA, if you add a big piece, it'll start making all kinds of proteins. There's no way to keep track of it. But if you add a very simple and small enough piece of RNA, you might get a sense of what the amino acids are for that piece of RNA, and you might be able to crack the genetic code. And so Marshall Nuremberg is quite excited about this. He's so excited is that he flies from his lab, the NIH in Maryland, he flies to California, and he meets up with a very impressive scientist studying at the University of California, Berkeley, Wendell Stanley. You might be familiar with Wendell Stanley, who was working with tobacco mosaic virus. And tobacco mosaic virus is a simple rod-shaped virus with a protein code and a single strand of RNA. And so, Mark, you can